Okay, hi guys, welcome to Biology Online with Henny van Vieren.
Okay, welcome back to another exciting episode of Past Papers with Henny van Vieren. Anyways, this is question 2, and it's all about population genetics. Since the arrival of humans 700 years ago and other mammalian predators, the population numbers of most of the native species of birds found in Aotearoa, New Zealand, have decreased significantly. Now remember, before humans came here, the only thing that was here that was a mammal was a bat, and it's because the bat could fly here. And then we have only a very few species of lizard, and they would have arrived here on floating logs and that type of stuff. And then we had 0 other mammals. So the kakapo doesn't fly because it didn't need to fly, because the only predators here was the Australasian harrier or the New Zealand hawk, and also the New Zealand falcon, and then the horned eagle. So where did the kakapo live? It lived in the dense forest, and that's why it gave up its ability to fly, because it's easier for it to just walk around and there's nothing hunting you. And also the issue then was, when humans arrived, they brought along, the first humans who arrived here, they brought along rats and they brought along their dogs. And that was a huge problem for these birds, because they had no fear response, meaning they didn't know to be afraid of these things. And hence why their numbers, the total number left of these birds, is 211. So, in 1977, the last wild population of kakapo was discovered on Stewart Island. The bird's main predators there were thought to have been feral cats. Feral means they used to be domesticated, but they became wild again. So there you go, there's your domestic cat. And if you have a cat, it has certainly brought you some trophies. That's what they do, they're predators. We can't blame them for that. But if we know that we need to protect some other organisms, the best way to prevent it from catching birds is put a bell around its neck. Then it can't sneak up as well. Okay, it's very simple. So, in New Zealand, there's 211 kakapo and about 1.419 million cats. Wow. And if you look at genetic diversity, obviously the kakapo is going to be very low because of a low population number. And cats is going to be huge because we imported them from all over the world. They are not just cats from, I don't know, South London. We brought them all in from all over the world. So, using the information above, discuss why genetic diversity of the kakapo is low and justify why the cat predation, that means killing, has resulted in genetic drift. In your answer, discuss genetic diversity and allele frequency, an explanation of population bottleneck, and why small populations most likely have low genetic diversity, explanation why this is an example of bottleneck effect and not founder effect, and a discussion why the kakapo predation by cats has resulted in genetic drift. Okay, here we go. Now, first thing, genetic diversity. Genetic diversity is when we have a very high number of alleles for the same thing. Okay, so in cats, for instance, there's lots of different color cats, okay, and also different sizes. And I think about Siamese, I think about Persian cats, I think about, what's that big one, I think it's called a Mancoon or something. All these different types of cats, and then within those you have different colorations and so forth. So that is a very high genetic diversity. Whereas a low genetic diversity means that there is a low number of alleles within a population. So, and allele frequency then, if you have a high genetic diversity, your allele frequency is going to be low, because what you do is you take your number of alleles, or that how much that allele actually is present in the total number of alleles. So let's say the allele for a blue-eyed cat, there could be 2 in the whole of New Zealand. I'm just making this up. And so that's going to be 2 out of potentially 500, which translates to a very low percentage. So you do that and you multiply it by 100, and that gives you, so it is your the amount of times the allele appears in a population divided by the total number of alleles. That is allele frequency. Okay, the number that your allele appears versus the total number of alleles present. Okay, so in the Kakapo, even if every single one of them had a different allele, the maximum number of alleles will be 211. In cats, I mean, even if they have, I don't know, just because of the simple numbers, it is way higher. Okay. Now, then, a bottleneck effect. So what is bottleneck? Bottleneck is when our population went from a very diverse or a high genetic diversity, and due to a natural disaster or even an unnatural disaster, but some kind of disaster brought it down, and it then ends up with a very low genetic diversity. And in this case, it was the arrival of cats, which basically wiped out the population of Kakapo to only one group of individuals left, and consequently the amount of alleles that were lost in comparison to the original population will be incredibly high. Okay, and then why small populations have low genetic diversity. We just said that because we lost all of these alleles and the ones that died due to the bottleneck effect. Can you see this is a bottle that's upside down? And there's the cap. Okay, so all your alleles, they come in here, but unfortunately only 1 or 2 of them will come out because of this cat. Okay, then explain why this is an example of bottleneck and not founder effect. The reason why it's bottleneck and not founder effect is when you have when a small group will emigrate or move to a new location. Now that is not what happened here. What happened here was the cats arrived, and the cats might have experienced founder effect initially because there were a small number of them. But humans being humans, we brought along a lot of different cats, which means that we artificially increased the genetic variation amongst them by bringing in more and more over time. Whereas the Kakapo, we didn't move it to Stewart Island, it was discovered there, it survived there. So consequently, everybody else was killed except those few, so they were survivors. Survivors is not the same as founders. Founders are when they move to a new area. Okay, and then a discussion of why the Kakapo predation by cats has resulted in genetic drift. Remember, genetic drift is the random movement of genes within a population, not due to natural selection. And the reason for that is that with such genetic drift has a very large effect on small populations. So if there's only 211 and there's in that 211 you have 4 of them that pass away, there's quite a few alleles that are going to be lost. So the effect on small populations is way higher than in genetic drift, and also because there's so few potential mates out there, you will then spread genes not due to natural selection but simply by chance, and that is genetic drift. Okay, so that is a nice simple and short question. Let's look at the model answer. Okay, so question 2, genetic diversity is the variation in the genetic makeup, the total number of genetic characteristics or alleles in a species, population, genome, or then gene pool. So you described what genetic diversity is because I told you to study your definitions. Describe the allele frequency. Allele frequency is the number or the amount, percentage of each allele in the population. So the percentage, that's why I divided it. And we studied that. A population bottleneck is the sudden, rapid, drastic reduction in the population numbers that result in an underrepresentation of the alleles in the gene pool compared to the original gene pool. Yep, you studied that. Beautiful. Founder effect, and just like that, you've already passed. And all you did is write down definitions. Founder effect is when a small group of individuals from an existing population moves to another area and establishes a new population. They are usually isolated from the original population and will have a much lower genetic diversity in comparison to the original population. Yep. Genetic drift is the random change of alleles in a population that results in the loss of alleles or some alleles becoming fixed. And yeah, so that's all they wrote there. Low genetic diversity means low number of different alleles in a population. Some populations mostly likely have low genetic diversity because there are simply less individuals that can have different alleles. So it is more likely that the allele frequency is not representative of the original population. There is an increased likelihood of inbreeding. Small populations are also more likely to lead to fixed alleles or death as larger proportional effects decrease. So in other words, if you can lose an allele very easy or you can fix an allele very easily. Then, predation by cats resulted in genetic drift because they have randomly quickly removed a large number of individual Kakapo from the population. Always remember to refer back to the example. If you just talked about predation and you didn't mention the cats and you didn't mention the Kakapo, then you cannot get your E, you can only get an M. Okay? The Kakapo have not been able to increase in numbers because the population of predators has increased very rapidly. They have also evolved in a predator-free environment, so they had a lack of adaptation to avoid predation. The Kakapo population has been reduced due to the indiscriminate predation by the cats and has resulted in only 211 individuals in New Zealand. However, the founder effect is when populations become reproductively isolated from an original group. The new population is not representative of the original group. Because the current Kakapo does not have an original population and its only population in Aotearoa, hence this is a bottleneck and not a founder effect. I think we did that relatively well. So for merit, you explain why the small population have a low genetic diversity, you explain genetic drift, you explain why the Kakapo has not been able to increase in numbers, and explain why this is bottleneck and not founder effect. And then if you did a very good discussion of genetic drift and correctly used the context, see there, context of cats in the answer, you justify why this is an example of genetic drift, discuss how these links, and why an example of bottleneck and not founder effect. Easy peasy. Okay, so look here, how many A's do we have? 1, 2, 3, 4, 5, 6, 7, and 1, 2, 3, 4 M's. So to get an M, you only needed 3 out of 7 A's. You needed 2 out of 4 for an M, 3 out of 4 for an M plus, and then discussed one criterion for excellence and both criterions for excellence. Easy. That's actually a really easy question, I would say. Okay, so again, I'll stop the video and then I'm going to do question 3.